All right, good morning again. Um, the Today we're focusing on lecture 5, and it's the principle of microbial molecular biology, and we will discuss genes, gene expression, and all the related subjects. Okay. As an introductory, this is a figure that you probably, or it's called the central dogma, that you've heard probably since high school. And so genetic information is carried in the DNA, and then it gets replicated. So that it can be passed from one generation to the next, or from one cell to two cells in our case, when we think about bacteria. But it goes in the other direction, is that the genetic information is actually being used to make protein. Okay, so it flows this way as well. Because the proteins are the actors that in the cell that get things done, and those are very important. And in addition, the gene arrangement in prokaryotes and eukaryotes are different. So in prokaryotes, the genes function in one particular pathway are often linked together, because the proteins are required at the same time. Okay, the way you think about this is that if you have a pathway, so the famous promoter that will be, or the operon that we'll be talking about, the LAC-Z. So in order to use lactose, you need at least two different proteins. Missing one wouldn't work. So if you were to make a protein for using lactose, you need both. And so that's why the two of them are often together, coded in the same region, transcribed at the same time. And the transcription just generates the mRNA. mRNA goes directly into translation. But for the eukaryotes, that is a little bit more complicated. So a gene is actually interrupted with intron and exon. So I'm sure you already know this. And you have exon and you have intron. And the introns are not expressed because when it's first transcribed, you have a primary RNA, and then the RNA gets processed, all the intron gets removed. Then you have a mature mRNA, and the mature RNA gets transported to the cytoplasm and then gets translated, and the protein gets made. All right, and so that's usually the difference. So the intron is something that's unique to the eukaryotes. Now, that's a quick overview, and those are the important issues. And so for the next, the rest of the lecture will be focused on two parts. One is look at the gene, and then we look at the gene expression. To look at the gene, we basically have to first look at the molecule that actually carries the gene. And then we look at the DNA replication. All right. So for DNA, we first need to look at the DNA structure. And so DNA typically are presented in two different ways: the ribbon format or a filled-in model. So this model shows all the sugars and backbones. So these are sugars and phosphates, and then the part in between are the the base pairs, the base. So, and then it could be also represented in the ribbon format. And the ribbon format is really just only showing you the backbone. Okay. Now, you've seen this before. There are two things I wanted to point out on this, this particular slide will become very important later on, is this concept of major groove and minor groove. All right. And then the other is this 3 and 5 prime. Those are two sets of very important information. So first, I wanted to discuss about the major groove versus minor groove. But what is the minor groove pointing out this part? The major groove point out this part. So the first thing you think about, the difference between major and the minor is, well, minor is smaller, right? And so it's fitting that it's smaller and it's larger. Okay. Now, if you were to grab this molecule, and where would you grab? Would you try to grab in the major groove or the minor groove?
Major.
It would be easier to, yeah, it would be easier to grab the major groove, right? Because it's bigger and your hands gets in really better. Okay, so and that actually is true. So the difference between functionally the major groove is where the protein will bind. Later on, we'll learn that there are proteins that regulates the DNA and interact with DNA, right, to get the thing out, get the genetic information out. And all interaction is through the major groove. Very, very rarely through the minor groove. Okay, so that's the major groove and minor groove function. Now, I want to go and explain what is the difference between major groove and minor groove. Okay, so this, you see this here, let me just use my pen here. This, you see the line I draw? That represents what? That represents one base pair, right? This, maybe I should change color. Wait, change color here. Okay, so you have one base pair, next base pair, right? So this is one base pair. All right, so if you focus on this one base pair, then you can see the base from the yellow on one side, orange on the other side, and the backbone from the other side, right? So now keep that in mind, and then I'm going to switch to a different slide, the next slide. And so what this shows you is the cross section of that, the line that I just showed you. So this is the base pair, the base, right? The cytosine and guanine. And that's where the 3 hydrogen bond is, right? So it's a GC and allows 3 hydrogen bonds to form between the two bases. And so what I wanted to point out is where the backbone is. So the backbone, this is the sugar backbone. You see, okay, the sugar backbone. And can someone not stop talking on the phone? Sorry, I'm muting whoever's on the phone. Sorry. Um, so what I wanted to point out is you see the distance between here and the other side? Because the backbone is not exactly opposite to each other, then you ended up with this being shorter distance and that much more space. And same is in the AT base pair, right? And so this is, and that is much bigger. And so what is this? Do I type? Can I type? And so here is the minor. And then obviously here, well, I need to say, here is major. I'll type somewhere better. View. Okay.
So the
Major.
guanine one is minor. It's the small one. Okay.
So yes. So the smaller area forms the minor groove, and the larger area forms the major groove. So not only the space is there, but also the charged group. So proteins interact by by charging, right? How sticky they are. And so if we were to then go back to this graph, this slide, so now you see why this is a wider area and the other side is a narrow area, right? And so that's why this is the major groove. And so and of course the DNA stacks one on top and then it turns too. And so that's the major groove goes around and the minor groove goes around. And so that explains why you have major groove and a minor groove. So now you understand the function of the major groove is for interacting with protein. And that's the major groove. And then now you understand why you have this major groove and minor groove business. And now if you turn to the ribbon format, now you can see why is the DNA is drawing this way, right? So now you can see, okay, this is the minor groove and this is the major groove. You see the space here? That's what's representing. Okay, so that is something new. I'm hoping that you have not heard from anyone else, but that's important. That's the basis for DNA and protein interaction. So now I'm going to focus on this 5' and 3'. What is that? Anyone know what that number stands for? No, not cap and tail. The number has a numbering system. So I need you to think about organic chemistry. You all had organic chemistry, right? If you're not, then you do. I'm sorry, I'm joking. You'll be fine. So the number 3 whatever is here. Can you all see the number? Yes, the number is the carbon. Okay, so now you understand the the 5' is here. Where is 5? 5 represent the 5' carbon. That's the 5, and that's the 3, because it's 1, 2, 3, 4, and 5, and that's an oxygen, right? Okay, so the 3 and the 5 is referring to the carbon position. And the 5' is always the phosphate. The phosphate is on the 5', and the hydroxyl group is on the 3'. And since it's DNA, this is what? This is 2 carbon. So the DNA is actually a 2 deoxy, right? Because when you think about deoxy, DNA is a deoxy, and then people ask, what is, where is the deoxy gets oxygen lost? Well, it's lost, right? Okay, so if you focus on this, then you have the DNA is always drawn this way, and so you have DNA on one side, one chain is 5 to 3 all the way down, and so this 3, and this is 5 prime N.
So the deoxy
And
prime?
say it again.
You said the deoxy gets lost on the 5 prime?
No, deoxy gets lost on the 2.
On the
2
2.
deoxy.
Okay.
Right, because that's what it shows. Okay, and so here's the very important part. If you look at this, the complementary strand, the DNA complementary is the opposite direction, is 5 to 3 in the opposite direction. Why? Because that's the only way that can fit. Okay, so now you understand there is a term called anti-parallel, right? The two are anti-parallel. So the DNA, so whenever, so here's the very important rule you need to write down, or that will help you in the future, is that whenever DNA or RNA form double strand, either DNA, RNA, or DNA, whenever they form double strand, it's always anti-parallel. It's always anti-parallel. And it's always expressed using 5 to 3, 3 to 5. Okay, so now if you're back to here, now you understand a simple number means a lot. And this tells you the DNA strand is an anti-parallel. At one end is 5, the other end is 3 prime. Okay? So now back to the DNA structure that has, let me back here, is the DNA is formed by base pairs, right? And you have GC base pair and you have AT. And GC has 3 hydrogen bonds, so it's much more stable than AT. How stable you can actually measure, all right? And you can, if you have a piece of DNA and you can measure it, and if you heat it up, if you heat up the, if you increase the temperature, it gets hotter and hotter, and the DNA actually gets denatured. All right, so when you're at 72, and so from here, mostly not denatured, but when it gets here, it's all denatured. So somewhere in the middle is T melting point, and so the melting point temperature is 50% melting. And so obviously, the higher the GC, if this is all GC, the melting temperature will be higher, right? And the curve will shift up here. And right, and if you have all ATs, the melting temperature will be, the TM will be less. So it'll be like this, right? And the other one will be like this. Okay, so this will be then so this will be high GC, and this will be a DNA with high ATs, right? And so, if we back to our best friend or your best friend now, E. coli is your best friend, right? I assigned it to you. That you need to know a lot. E. coli, to give you a reference point, E. coli is 50% Gs, GCs, and 50% ATs. Okay, now you understand, okay, so a bacteria that has optimal growth temperature at 37 or 39, GC is at 50%. So if some bacteria would live in a hot spring, not super hot, but like in hot springs, 65 degree, and that bacteria should have higher or lower GC. Yes, thank you, Maria and Estefan. Yes, it will have higher GC because it needs to maintain the DNA, right? So that's related to GC, and that's GC content is important. Also related to the base pairing is that if you have these base pairs, and if you have this inverted sequence and in opposite direction, then when you separate, when you denature, and then when you allowed it to re-anneal, then you will form stem. So you will form stem and loop structure, because you see this and this G can form, can just be here, right? And so you separate and then they stick together. So it's almost like if you have two Scotch tapes, right? And if you separate the Scotch tape and you dangle them around, and they will stick together. And thinking about Scotch tape, this is even more like a Scotch tape. So if the end is complementary, you can have this one part fold back and stick together, and then form a hairpin structure. So this is not called stem loop. This is called hairpin. Okay, this is really like the hairpin. Also related to this then is you can have linear DNA with complementary single-stranded ends. And this is called cohesive end. Cohesive ends. Or sticky ends. And so what that means is, remember, the DNA itself is very, very thin and very flexible. Think about the thinnest strand that you have. So it's very flexible. So when you have this DNA, and it will bend easily, and so if you have what they didn't say is this is, can I write it? And this is, if you were to add, this is 5 prime, and this is, how to say it again? This is also 5, right? Prime. And this would be 3 prime, right? This would be 3 prime. Okay, so when the loop is wrong, and you will find that this G will match it with this C. You see, that matches. And so now they stick together. Okay, so this overhand sticky end, and they will fold back, and so this goes up here, this G finds this C, this T finds A, and C finds G. So now they stick together. Okay, so once they stick together, the linear DNA becomes a circular DNA molecule. This mechanism is very important. We take advantage of this for DNA cloning. So if you heard about DNA cloning, and that's what that really is. You can use this feature to add fragments to DNA. And in nature, viruses also take advantage of this mechanism, the sticky end, so that they actually have linear DNA in their head, the phage head, and then that's for bacteria. And then once getting to the host, they recircularize. Okay. All right. So that's about. Say it again.
You said that the viruses do this?
Yes, Lambda virus does this. Okay, so what we just talked about is the DNA structure. We introduced I explained the complementary and the anti-parallel of the DNA, nature of the DNA and RNA, and anytime they form double strand, because this is important when we think about transcription later on. All right. And then the base pairing and its impact, because base pairs, they don't face each other. They face in an angle so that they create the major groove and minor groove. So if you can see my hand, it literally like this, right? I should have 3 fingers, right? 3. So like this. Not straightforward. If it's a straightforward, then both sides is the same. You don't have major or minor. What happens in DNA is they tilt it, right? So that one side is the top is bigger than the bottom. And so that's where the major and the minor groove comes in. And then because of the base pairing, you have these specific sequence-specific features, melting point, stem loop, hairpin structures, and cohesive end. All right. Now, in addition, the DNA actually forms supercoiling. And so to understand supercoiling, basically, you would need to understand it's a double-stranded, right? So if you have a single-stranded break, remember the bonds can be the two molecules form bonds. They can rotate, right? These phosphoester bonds between oxygen and phosphate, it can be rotated. And so if you take one end and then loops around, right? And that's where you have you can form the supercoiling. Okay, so if you loops around, and you can actually form supercoiling. And I'm sure you've seen people play with a rubber band, right? So if you put a rubber band flat on the surface and you make them into a oval, and then you put both fingers on it, and then you rub it, what happened? I wish I have a document camera I can show you. So if you rub the rubber band, they will become a rod, right? And if you rub them further, they become ball, right? And that's exactly this the concept of introducing the supercoiling. All right, and so so this is supercoiling, and here are the so this is another different a ribbon view. So these two colors to highlight the complementary strand. So one, so it's a double-stranded DNA, right? And so you push one end to the other side, and then you break it over, so then it loops around, right? And so now you loop around, and so this is the top isomerase. So I'll address that question. So there are two ways to form supercoiling. One is to make one single strand nick, and then you have this special enzyme called top isomerase 2 that forms double-stranded break, and so that it can go in here. So then it forms double-stranded break and then reseal, and that also forms supercoiling. Okay, so that's that. So I hope addressed your question about the single-stranded break and double-strand break. And so they're carried out by different enzymes. So top isomerase 2 does the double-stranded break. Okay, so why do bacteria go through the trouble of doing that? And because I've tried to explain that your best friend will only do things that's absolutely necessary. Okay, so let me first show you what actually is happening. So this is E. coli chromosome. Okay, this is well, first of all, this is a picture generated by what kind of microscope? Can you want to guess? By now, you should be able to look at a picture and say, aha, I know this is pictures generated by a microscope. Of what kind of microscope? You are looking at the DNA, right? So obviously it's not the so it has to be electron, and it has to be transmission electron microscope, TEM. Electron is not sufficient. So I'm also trying to correct you by correcting everyone's. I'm also trying to push you to use a more precise description when you think about science and when you talk about science. So you can't say electron, and that's wrong because that's not sufficient. You need to say TEM, transmission electron microscope. Okay, so to be very precise, because electron could mean electron could be scanning electron microscope, but that's not that, right? Okay, so what the picture shows you is these, if you follow these loops, right, you can actually see there's a loop goes out and comes back. And this shows you bar. Where's the bar? The bar is 1. Okay, so it's 1 micrometer. And we know E. coli is what? Anyone want to venture? The size of E. coli? Is 1/3 micrometer. Thank you. Thank you, Estefan. Yes, you have, oh, I know I made you guys type. You can use that's an acceptable. So, because, so if you say, so 1/3, 3 and that is acceptable. Okay. So what then, because this is the bar, and so that means this is here and like this, right? And so what I drew is about the size of E. coli, right? So you see how much space is taken up by DNA. And now your challenge is to stuff all things in here. What would you do? Right? That And so what happens is that you need to stuff things into, I guess if you ever deal with the wire, electric wire or extension cord, right, like a 50 feet extension cord, you really have to wind them up really well so next time you can use them, right? And so what happens is that in order to pack the chromosome into the small space inside the bacteria cell, what they do is they use the supercoil, all right, and they form these loops. They form supercoil loops, and the loops are sealed by proteins, and these proteins are there just to make sure the loop stays supercoiled. Because if you think about the rubber band that I just described, if you loosen it, it's just rubber band will go naturally into a relaxed form, right? And so these proteins are there to seal the supercoil domain so that this remains supercoil. Now, in addition to helping packing, the supercoil also makes the DNA stronger because instead of instead of double strand, now you actually have two DNA strands together. So this is stronger than the DNA itself, right? And so if we go here, now you understand what's going on. So it goes out and comes back. All right. So that's how bacteria uses supercoil to pack its chromosome. Now, eukaryotic cells have similar problem of organizing its DNA, but they use different mechanism. They use core histone proteins, and this histone H1 proteins, and basically the DNA loops around these histones to form nucleosome. Okay, so I'm gonna mention, but I'm gonna ask you to do the homework of really understanding what the difference between genetic elements. And so these are your homeworks. So chromosome, generally referring to the DNA molecule in bacterial cell, carries essential gene. Plasmid, generally referring to a smaller circular DNA molecule that carries non-essential genes, and it's typically somewhere between 100 million base pairs. I'm sorry, 100,000, 100K. And let me just write it down. It could be 100 KB, all right? Kilo base pairs. Transposable elements are pieces of DNA, later on we'll talk about, can move from one part of the DNA molecule to another part of the DNA molecule. And viral genome, well, yeah, we'll talk about viruses have their own genome. And the reason they're here, we think about this, is that some virus can insert their genome into chromosome and becomes a part of the host chromosome. And you see this in bacteria, and you see this in eukaryotic virus too, like the HIV virus, right? The AIDS virus integrates itself into the host chromosome. And then you have mitochondria and chloroplasts, and we commonly think they derived from the bacteria, and eventually only kept its essential gene. And those are some of the concepts that you need to be aware of. Now I wanted to talk about DNA replication. So DNA replication is very important. The most important part, obviously, is the accurate replication. So the daughter copy will be the exact as the parent. So what they do then is the DNA is replicated through what's called the semi-conserved replication. So this is to show you that the parent in the light color, and then when replicated, one copy goes to one daughter, right? And so each of the daughter cell get a copy, and then a new one gets made. So that's the important concept. It's semi-conserved replication. Now, to understand the replication, you have to understand how new DNA is synthesized. So the bottom is the old. The bottom is the old. The top is the new. The new DNA is synthesized by the DNA polymerase. And the DNA polymerase requires template and primer. So it needs a primer, and in this case, it's the primer is a primer so that you can make new DNA, and it needs a template. So the DNA polymerase will only extend based on the template so that it will match what base pair is there. Okay, so now let's first take a look at the DNA polymerase, the function of DNA polymerase. So here is a DNA molecule, okay? And now if this is DNA molecule, this is the 3' end, right? This is the 3' end. Why is it 3' end? Well, because you have a free hydroxyl group. Okay, again, 2 is deoxy. Now, if we think about later on, we'll talk about the RNA polymerase will carry the same exact reaction, but to extend the RNA. The difference between DNA and RNA is this oxygen here. DNA does not have this. So when we look at this, this is DNA. And so the DNA polymerase carries out this reaction, this oxygen to this phosphate. And the energy for this reaction is carried out from this bond. This is where the energy is. So notice you have triphosphate. This is, so if it's T, well, we don't know, right? We could be applied. So you have ATP, you have GTP, you have CTP, right? You can have TTP. So these are the 4 nucleotides, triphosphate nucleotides, ready to be the substrate for adding or extending DNA. So this can be extended, right? Because you need energy for this reaction. So if you take this molecule, go on to here. So think about this, right? And you can take this molecule and move up here to this, right? To the phosphate. Say, okay, I want to form this hydroxyl group with this reaction. Would it work by the DNA polymerase? No. DNA polymerase will not carry out that reaction. It will only carry out this particular reaction. Is it clear? Anyone have a question? This is about the fundamental as you can get. And if you understand this reaction, many of the problem will be solved. Okay, so you basically need.
The DNA polymerase won't go with the one above it because that's not its primer.
No, the DNA polymerase, let me restate. The DNA polymerase will only carry out this reaction. How do I clean everything? Eraser. How do I do erase? Okay, I erase everything. Okay. Let me just restate what is the most important part. So you have the 3' end, right? We understand this 3' end. That's where the DNA polymerase comes in. The DNA polymerase only carries out this particular reaction. That's it. Okay. So now if you go back to this diagram, now you understand why this is true this way. The DNA polymerase will only add nucleotide to a free 3' end. So that means that because of that, because of this reaction, the DNA can only be extended at 3' end. It will not grow at 5' end. Okay. And why do you need an RNA polymerase? Well, because the DNA polymerase will only add to a 3' hydroxyl group, which means if the RNA is here, the DNA polymerase can still carry out that reaction. Okay, so now just to reemphasize, the DNA polymerase, the role of DNA polymerase is to add this reaction, carry out this reaction using the energy from the triphosphate. So if you have AMT, it will not be able to be incorporated into DNA. It has to be ATP. It has to have the triphosphate on it. Good? Anyone? Okay. So once you understand the DNA polymerase reaction, then you can look at how a circular DNA molecule is replicated. So it actually, so we're looking at a circular DNA molecule, and the first important concept is that it starts from a particular part. Okay, so this is start from the origin of replication. And on bacterial chromosome, there's actually a region called origin of replication. And you would guess it fills with, it's an area with lots of A's and T's. Why? Why would you need more ATs in the original replication? Well,
is that that's where
that's where the bonds are weaker, and so that you can separate, because this is the part that needs to be separated. That's important. Then the other part is that the replication, the directional replication, so that you can actually replicate this all the way through, right? But you can, what actually happened in E. coli is that they do this. Okay, they do bidirectional replication. You see this? They're replicating it at the same time from both directions. And so that shortens the replication time to half, right? So that makes it faster. So let's take a look at what's actually happens in the original replication. So then you have the replication takes place in both directions. Okay, so this is called a replication fork. Okay, this is important. So in the replication fork, you see you have the, if you focus on this one, or you focus on this one, either one is fine. And this is what's called a leading strand replication, and this is called a lagging strand. Why is a leading strand? Well, because this is extending at the 3' end, right? Because it's only extending at 3' end, and the 3' end can go hold your question. So this is the 3' end, and it goes in the opposite direction. Notice this one is a 5, right? So it's anti-parallel, goes in, and the top is 3, and this is 5. It's complementary, right? And so this is actually, I will come back to talk about how this part is replicated. So this is easy, right? And you see this goes in, but what about this part, right? Because it's only extending on this side, not on that side. So we'll come back to talk about the replication fork. But for now, we understand its replication takes place in both directions. And the replication requires the DNA to be unwound. And so the helicase does that job of just unzip the DNA. So putting together for a circular DNA replication, what happens is that you separate and you replicate at the same time. And when it's completely separated, and the two are completely replicated. Okay, so now I wanted to come back to talk about the replication fork. What's happening in the replication fork? So we talked about the leading strand. Remember the leading strand that was shown, the leading strand. Well, if we focus on leading strand, that's 5, and that must be the 3. So we have a 3 free hydroxyl group, right? So the DNA polymerase is very happy. Just get a nucleotide, a TTP or CTP or GTP, and they just put it in, and the helicase keep unwinds the DNA or separates, and then you have some single strand protein binding to stabilize the single strand DNA. Because single strand DNA is very, very fragile. Okay, so that's straightforward. Now, the harder part is to understand the lagging strand replication. Why? Well, because the lagging strand, because you still have the template, is 3 to 5, right? And so 5 is here, so this is a 3' end. And so the direction is going there, and it's going away. So so now what happens is that you actually have to prime it much more often. So the primase, which is RNA polymerase, well, comes here and lay down and reads the DNA and put a stretch of RNA in there, and then forms the primer. Okay, so the yellow primer. And so now this then the RNA polymerase comes in and then go all the way here. All right, and go all the way here. Then what? And so if we were to go to the next pictures, you will see the DNA polymerase can come here, but it will then remove the primer. Okay, removes the primer, and then it needs okay, so the DNA polymerase 1 comes in and removes the removes the primer and fills the gap. Okay, so the DNA polymerase here fills the gap. Now it's between 3' hydroxyl group and the 5' phosphate. And then the DNA ligase, that's where the DNA ligase comes in. So the DNA ligase then fills the gap, which they didn't say here, the DNA ligase and ATP. The DNA ligase needs an ATP. Why does it need ATP to work? Well, because that needs it needs the energy. Right? Remember, when you form a bond from your chemistry class, right? When you form a bond that has energy, you need energy to form the bond. So that's where the DNA ligase comes in. So now you have a complete DNA, new strand of DNA. So this is what's happening on the lagging strand. All right. So now we actually introduced a concept of DNA polymerase 1 and DNA polymerase 3. DNA polymerase 1 is actually a protein complex. Okay, so this is actually the DNA polymerase 1. And so it actually is much more complex. And then it has DNA polymerase 3 unit, and it's involved. And okay, and so now the forms are complex, and then the DNA goes in one strand. Okay, so the DNA. So DNA, the parent DNA, and the new daughter DNA comes out. So the whole complex is referred to as replisome. Okay, so replisome, and the replisome forms a protein complex and allows the DNA to zip through, to go through, and it's really fast. All right. So that's the DNA replication, and related to this important concept is the DNA polymerase. I'm sorry. Let me go back and make sure I didn't misspeak. Okay, so the replisome, and this is DNA polymerase 3. All right, we want to highlight polymerase 3. We also had in this slide, we say DNA polymerase 1, DNA polymerase 3 does this, and the DNA polymerase 1 comes in here and removes and then fills the gap. Okay, but in the actual replisome, this function is carried out entirely by the replisome. Okay, so when N comes in, when N goes out. The DNA polymerase 1 is actually you guess the DNA polymerase 1 is the one that was discovered first, and that the DNA polymerase 1 is actually one protein, and 3 is a complex. Okay, so 1 is one protein. It was first isolated. It's actually interesting by one of the giants in the molecular biology called Arthur Kornberg, and he studied the DNA replication, isolated DNA polymerase, and then he found out the DNA polymerase 1 is not essential for replicating chromosome because he can delete it and bacteria still grows. And then until his son, Arthur Kornberg Jr., joined and grew up and start studying DNA replication, and then he found out it's actually, well, his son, Junior actually found out that DNA polymerase 3 is actually a protein complex. So that's an interesting story. So we then back to the question of why is the DNA polymerase 3 so accurate in replicating? Does it ever make mistake? Well, it does make mistake. What the trick is that when it makes mistake, it finds out immediately. Well, the way you think about this whole thing, DNA replication, is, well, if you have 4 bases and that's in a bag that you have to pick out, and they feel very much similar, right, in a bag, and you have to pick out, then often you will make a mistake. But when you actually, when proteins work in the dark, and what they do is if they find that it doesn't form the correct match, and probably the shape and size is going to be different, right? And that's where it stops. Okay, so now the DNA polymerase 3 had a mismatch, A and C, what puts it in the wrong. And so then what it does is it already formed this bond, right? So, but it also has an exonuclease activity. So exonuclease. So nuclease is an enzyme. Exo means it from the end, from the outside. This is in contrast to endo. So you can have endonuclease, right? The exonuclease cuts from the end in one by one. Endo just cuts in the middle. Okay, so the DNA polymerase 3 actually has exonuclease activity. And that exonuclease activity is opposite from the polymerase activity, so that it is from 3 to 5. Why? Well, this is 3N, right? This is 3, and this is 5, right? And so it goes backwards. So now you understand it can go back and cleave, and then try again until it finds a new nucleotide and correct, and then move forward. Okay? So that's how it's replicating. So that's how it's replicating the circular DNA molecule. Earlier, there's someone asked about replicating linear DNA molecule. Now, bacteria, majority of the bacteria have circular chromosome. And replicating linear DNA is something much more complex. That's more like us, the eukaryotes, have linear DNA. And that requires telomere and other complex proteins involved. And so for bacteria, mostly we talk about replicating a circular DNA molecule. In fact, if the bacterial chromosome gets linearized, bacteria will die because the linear DNA cannot be replicated by majority of the bacteria. So if you think about E. coli only replicates circular DNA molecule. E. coli does not replicate linear DNA molecule. Okay, so that's the DNA replication. And so with the replication, the match, and the repair, it ensures the accurate DNA replication. Now, the DNA is accurately replicated. It only useful. It can be used to make things, right? And to make proteins. So let's take a look at how it's used to make proteins. So there are two major steps, transcription and translation. This is a outline slide. And when I say, well, after you review the class, when you look at the word transcription or the word of translation, you should have a whole series of pictures flashing through your head. It's like, how does it actually work? You need to understand how each of the process work. Okay, so here is the transcription, how it starts. So the transcription, so first of all, you have, if you look at this, the DNA molecule, it's double-stranded, so it's 5 to 3 and 3 to 5. And then you have the actors, so the RNA polymerase, or it's called a core enzyme. And then you have sigma factor. The two of them combined together actually get onto the promoter region and then start the transcription. And it specifically tells you the light green strand is being transcribed. Okay, keep that in mind. And so what's important is that the sigma factor is the part that recognizes the sequence. Okay, so what does that mean? It's, if you focus on here, you will see this is the part, this is sigma factor. And sigma factor binds DNA. It binds to major groove or minor groove?
Major.
Yes, thank you. Major groove. And it has two binding sites. Okay? It has two binding sites. Just imagine that I am, hold on, if I jump onto the monkey bar, I want to have two hands hold on to it very tight. Okay, now here's again the monkey bar analogy comes in. You see, it binds, these are the, so from 1 to 6, these are 6 different promoters. 6 different promoters. And what's in common is this region, the binding sites. Okay, so you see the sequence is the same. What's also very important is the distance between them are the same. You see, the sequence is different, but this is the highlighted conserved sequence is the same, which means what? So if you imagine I am the sigma factor, I need to bind, I need to jump up, right? And but if I'm a sigma factor, my hands are just like this. It's not flexible, right? Proteins are not that flexible. It's like this. So I jump up and hold on to two. If I find 2 monkey bars, I can hold on to it. Now, what happens if some protein promoter have one binding site further away? That means what? If you move one of the monkey bars, I jump up, I grab one, but I can't find the other one. What happened? Well, if someone is stronger, I'm sure in our class we have stronger person hold on to the monkey bar with one hand, right? But majority of people is like me. It's like I need both hands. If I only got up one, I'll fall off. Okay, so the sigma factor recognize the the promoter by binding to a specific sequence. If a promoter doesn't have a sequence that's recognized by the sigma factor, then it will not bind. So now we're back here and say, okay, so the sigma factor recognizes where the promoter is and binds to it. And now, and then the RNA polymerase starts the transcription. And notice, what is this? This is new RNA, right? And remember what I said about DNA polymerase can only extend at 3' end, and RNA polymerase is the same. It can only extend at 3' end, right? And so this is the 5, and here is the 3, right? 3 hydroxyl, so it's extend. And so now the new mRNA is read. Okay, now we're back to the question about which strand is actually being read. It says the light green. Why is light green? Well, because if you think about it, remember the rule I mentioned earlier. When you have double-stranded DNA or RNA, it needs to be anti-parallel. So you don't even need to know which direction the template is. If you know this is 5, and it must be forming or must be reading the anti-parallel strand, right? So it's 5 to 3, right? So it's 5 to 3, and that what a strand is being read must be 3 to 5. Okay, and so this can be read in here. So this is the double-stranded DNA, and it's being transcribed. And let me just use the next slide. And so just to highlight that you have to transcribe two strands, right? That's the DNA. And just to highlight which strand gets transcribed, the mRNA actually has the matching sequence of the 5 to 3 strand, but it forms anti-parallel with this strand, the 3 to 5. So we know that this strand is being, we know this part, sorry, the bottom, right? This, you see, this forms exact anti-parallel with the new RNA. So that's where we know it's being transcribed. Okay, so that helps you understand. This slide helps you understand which is being transcribed. So now we're back to here, the whole process. So you have transcription starts and then ends. Wait, how does sigma factor, how does RNA polymerase know where is the end? If you read the DNA, will you be able to figure out where's the end of where do you supposed to stop? Well, okay, here's where this, no, stop codon is for translation. It is extremely common to mix transcription and translation. To stop, for transcription to stop, this is how it works. If remember the stem loop structure, yeah, when you have single-stranded mRNA, it will form easily stem loop. Why? Well, think about this. If you get, you all got Scotch tape, right? You think about if you have a Scotch tape. If you draw the Scotch tape very long, this long, you don't do that. But if you draw it out and then you dangle it up a little bit, you have to be very, very careful, right? Because you know if you don't handle it well, it's just gonna go stick up, right, to itself, and then forms these wrinkles. And that's the same thing as a stem loop structure. And what's unique of this is that where is RNA polymerase in this whole thing? Well, the RNA polymerase, let me just draw it out here, is here. So that's where the RNA polymerase is. So it's the analogy is like if you drive, well, I assume most of you drive, you see a stop sign, you stop, right? But this RNA polymerase has a rear view mirror. It just see after it drives, after it's synthesized, does it form stem loop structure? If it forms a stem loop structure in the DNA, in the RNA, it just synthesized, then it's the signal for it to stop. Okay, so almost as if the stop sign is at the back, and if you drive, and you all of a sudden see, oh, a stop sign pops up on the rear view mirror, and you that's where you stop. But obviously, don't do that. I'm not gonna pay for your traffic tickets if you do that. Okay, that's okay. So now is it all clear how it starts and stops? I'll stop for a minute so that we catch us on. So transcription produces mRNA, and we need to know who starts it, who's the actor, does the transcription with RNA polymerase. Where does it start is the sigma factor. The two of them together starts the transcription. Once the transcription starts, the sigma factor actually leaves and no longer needed, because it's the only job is to determine where to stop. Where to start. I'm sorry, where to start the transcription, and then there's a stem loop structure start forming here, which is not drawn here. Stem. Let me see. A big stem loop here, and that's where the stop is. Okay. Now, when you actually view when you actually take an EM picture of a living cell, you can actually see transcription happening along the DNA, but it's actually interesting. So here in the middle, this line is DNA. Okay, and then if you look at here, you will see this line, right? This line. Okay. Now these black dots, okay, you see one here, and it's longer on this end, shorter on this end. And so what that is if you come here, this is what's happening. You have the RNA being transcribed, and then the mRNA, once it's transcribed, it's immediately become available for translation. So so now you see this is here. So you see this has longer, right? This mRNA is longer because it transcribes all the way from here to here, then mRNA is longer. Longer mRNA has more ribosome. And these individual black spot is actually ribosome. Okay, so that means, that tells you the transcription and translation are coupled and synchronized. And why? Well, because remember, bacteria generation time is 30 minutes, right? They only have 30 minutes to make a whole new cell. So they need to be very, very efficient and very, very fast. How fast? Well, they couple everything. Okay, so so now we'll come back to here, say, okay, now you have transcription, we'll make proteins, we'll translate. And so that's where the start codon and stop codon comes in. And so start codon is here and stop codon is here, right? So then the protein gets made. So you need to understand that, okay, within each region. So when we think about translation, so this primary, okay, so this is again transcription, and then so this is promoter, this is on DNA, this is RNA, and there are some interesting exceptions to the general rule, is that how the ribosome RNA is made, and this is a diagram specifically to show how ribosome RNA is made, and there are spaces, so you have RNA, primary transcripts, and that gets processed, and spaces removed. And so this is almost like the intron, but it's not. So this, then you have different species of ribosome RNA and transfer RNA, right? And so you have three different, for bacteria, you have 16s, 23, and 5s ribosome RNA, and then you have tRNA. Okay, so here is, again, I'm back to the synchronized, the transcription. Remember the picture I just showed you earlier? Remember this picture? Okay, and so if we zoom in and see that again, you have transcription and translation coupled inside a cell. So on this little black dots are actually ribosome. That's become that's visible. So this is the mRNA from 5 to 3, and this is ribosome reading or translating from 5 to 3, and this is peptide will come out, and it forms secondary structure between different amino acids, right? And so the initial, so here's the translation also involves where to start and where to stop, right? To start the translation, that what we call initiation. So this is the initiation complex. It has the ribosome binding site on the mRNA, so ribosome knows where to bind. This is the 30S unit, comes in and binds to the RNA, and then the tRNA, the initial tRNA comes in, that carries the methionine. Now this for bacteria is always the rule. So the first amino acid is always methionine, okay? Because the first codon, start codon is AUG. And then once it gets in, it needs a GTP to move, to form this complex. So now it's a complete completed initiation complex, right? So to putting in perspective, you have translation, initiation starts here. Once it starts, then it can go to loop. Every loop, so this is incorporating one amino acid. Okay, so this goes in, and this moves. So uses one GTP, moves to form the link, and then moves, and so GTP, and then goes to this empty, the empty tRNA gets removed. All right. So it's the ribosome. The way you best think about this is the ribosome traveling along the mRNA, and each time when it moves, it requires 2 GTP. So it requires 2 GTP to incorporate one amino acid. All right. And so this is how it translates. So if this is the mRNA, and this, if this is the correct sequence, it's reading 3 bases at a time, right? So go to ACA, it's reading, terracing, and that's how it reads. But if you're reading incorrectly from the A, right, that's a -1 frame, because you go backwards, -1 frame, you have a different set of amino acid. But if you go to the incorrect 1+ frame, you also have a different set of amino acids, right? And so this is actually a very important concept. It's called the reading frame. And this determines how fast or how accurate the reading is going. And so, and of course, to understand which codon is for what, and that's where you go and read. And so obviously, this is the genetic codon. You have the star codon and 3 stop codon. All right. Now, every semester, I always attempt to encourage everyone to remember the genetic codon. Remember this table? Anyone object to remember all of this table?
Do we have to?
Only one person object. I'm surprised. No, I'm joking.
My school, so I don't even
No, yeah, no, obviously no. You should all object to remember this table. Why do you have to remember this table at all? All you need to remember is the start codon and stop codon. Okay, so I'm just highlighting the complexity, and but it's interesting to read. So you can see there are 4. So there are 4 different codons for leucine, and most often you have 4 codons for one amino acid, sometimes 3. Now, bacteria, your friend E. coli, have no eye. Obviously can't read the table. Then how does that work? Well, how does E. coli accomplish this reading? Well, of the codon? Well, it relies on the transfer RNA, the tRNA, especially the tRNA with the anticodon. So every so you have 20 amino acids, right? So you have at minimum 20 tRNA. So one tRNA has one anticodon that matches the codon. Okay, it is very important that you notice the anti-codon and codon match is in the anti-parallel position. So this is 5 and this is 3, right? So just gonna say this is 5 prime and this is 3 prime. Okay, and so this is how bacteria ensure the correct tRNA, which is the codon. But what about amino acids? Who ensures the amino acids on the tRNA is correct? Well, that's actually carried out by a different enzyme. So the tRNA synthase. So the tRNA, this is to charge, so this is tRNA. To put an amino acid onto a tRNA is actually carried out by a particular enzyme. It actually consumes 1 ATP to charge the tRNA. And so this is the blue is the enzyme, the red is the tRNA, and the amino acid goes up there. And so now you understand this is all very well controlled. So the tRNA gets charged, put on a correct amino acid by an enzyme, right? By structure matching. So now you understand to add 1 amino acid, you actually require 3 ATPs. Okay, so that's how expensive that is. So you are then comes to the Wobble concept, and what the concept is, well, it tries to say is the codon. For the codon, the first 2 are very important. The third is flexible, and so allows the matching of a less frequent matching, because remember you have 4 different codons. Maybe a tRNA one tRNA can match 4 different codons, right? And so this is where the wobbling concept comes in. So that's the transcription and the translation. And after the translation, the protein forms. And so when protein comes out of the ribosome, when the peptide comes out of the ribosome, the amino acids interacting with its neighboring amino acids, right? They come out in a sequence, and depends on the location of the amino acids, they form different types of interactions. And so then they can form secondary structure. The basic secondary structure is helix and base. And this is all determined by the sequence of amino acids. Now, obviously, sometimes things go wrong. Proteins gets misfolded. And then what? So if you have a misfolded protein or improperly folded protein, then what you do is you put it through a molecular chaperone. Essentially, think about this as a ring, and you pull this through, and the amino acid, so the peptide comes out, and almost the same way as coming out of the ribosome, right? It comes out one by one. So then allows the amino acids to interact with its neighbor in the correct sequence. And that's how protein is then regained the correct folding. All right. So that's the then the protein is folded correctly. All right. So what we just talked about is how genetic information is being used to transcribe into mRNA and translate into peptide, and then peptide folded correctly into protein, functional protein that has enzymatic activity or other binding activity. It has unique structures. And if the protein was folded incorrectly or being disrupted by something, and then the bacteria has a rescue mechanism to use protein chaperone to fold it correctly. Okay, so that's so this is the slide that you should all use as a way of examining yourself or outline. Say, do I understand transcription? Do I understand translation? How translation starts, how transcription starts, and they are all very different, and you need to understand all of it. Okay, this is about as fundamental as the rest. And so understanding this will lay the groundwork for gene regulation and the other two other lecture that's to come following this. So I would urge you to understand, to review this particular lecture as soon as possible. Definitely before next Tuesday, so that when next Tuesday comes, you will be able to follow the regulation. So the next 2 minutes, I wanted to comment on the exam. So I've graded all the exam, and I will post the exam soon after today's lecture. So I wanted to give you the somewhat from your point is unexpected news, is that the exam grades are much lower than you probably originally experienced. But it's still within historical norm of this class. By that, I mean this this class, when I've taught many years now, more than 12 years, and it's the first exams, everyone always gets the lower grade, and this has many factors. One is really a complex subject to study, and obviously it's very complex and demands a lot more time, your time to study. And that's why in the first quiz, I specifically ask you how you plan to study. And I collect advice from previous students on how to study. But because, and I understand this is also a very, very even more challenging time than usual. We don't get to see each other face to face. And so this has become much more challenging. So I wanted to set up a time for us to discuss individually. All right. Let me first stop the